Good day everyone. Welcome to the course on biological data analysis and visualization with R. In the last three classes, we have talked about principal component analysis and we have done some hands-on analysis for principal components, and probably we are getting some idea about the principal component analysis and what kind of inferences that you can draw. So, we will continue that in this class. So, first we did principal component analysis using PRComp. This is a built-in function in R. Followed by that, we did principal component analysis using Factomine R package, and in both those cases we have also visualized the results that we got from principal component analysis. Subsequently, we then used the PCA tools. In the last class, I have introduced the PCA tools and different functions that are there in this package, and we have also introduced a new data set on which we are going to apply these PCA tools. Okay. So, we will continue that in this class. So, this is the agenda for this class. We will be talking about PCA analysis with the package PCA tools. Here is the link to the package. In the last class, we have seen the bioconductor page and the manual and some of the descriptions of this package. The data set we have introduced is an yeast noise data set where we have gene expression noise in yeast as well as other features that are there. Along with we have the metadata, right. So, this metadata included the different classifications of genes into different groups. Okay. We will again just very briefly show this see this metadata and kind of just to remind you, right, what this metadata looks like, what the data looks like, and then we will go into the PCA analysis, jump into the PCA analysis. So, this data is formatted for PCA analysis specifically so that PCA tools can take this data and do the analysis. We also have some of this metadata information that we can apply later on for visualization. Okay. So, let us now jump into the analysis, and let us remind you first about the data again very briefly and the metadata, and then we will go into the complete analysis. Okay, so, here is the R console and the data that we have, the data that we loaded is this one, right. So, this tab it actually has this genes along the columns, and we have different features as along the rows, right. So, what we are trying to do is we are considering genes as samples here, and we are trying to explain the variation that we have in the data in these features, right, using these features. And what we will get, right, what will be the outcome is finally how these features are related to each other. This is something we will be able to see from principal component analysis, and then we also see which features kind of contribute how much to the principal components. Okay. So, this is something that we will actually look into, and this is kind of a much more complex analysis than what we did with the cancer data set. Here we will see like we kind of need to understand what is happening at each step. Okay. So, let us do the okay, we have looked into the data, and just to remind you about the metadata, here is the metadata again. The genes are grouped into different classes, right, and these are names in groups, right, group E, H, F, etc. In the last class we have seen these are the different labels starting from group A to group E, group D, and then we have group E H, group E M, group E L, right. So, for group E we have 3 classes, and then we have group F. Okay, and then we have X for genes that are not classified into any of these groups. Okay, so, with that background, let us now move on to the principal component analysis. We are using this library called PCAtools, and we have done the first analysis, and as I said, maybe we do not need to do the centering of the data, we can simply work with the data. This is something you can also explore further, right. What happens if you do not do the principal component analysis, if you do not do this scaling, right, while doing the principal component analysis. Okay, this is something you can check, and whether how the results will change if you do both scaling and centering, and if you do only one of them, right. So, this is something you can explore, and kind of you will get a better idea of what actually happens, right. So, this is something that is required, otherwise we might not get the best results, because the features that have the highest values will often overload the PCs. Okay. This is something that is why we need to scale even if we are not centering. So, scale is absolutely necessary, otherwise you will see how these results will pan. Okay. So, this is something which we discussed in the last class. We got the PCA, and here we saw different parameters that we can access from P, right. P is the results of the principal component analysis. So, P rotated is that the values in terms of the transform data, right. When you transform the original data in terms of principal components, so what would be the XYZ values if you want to visualize them. We looked into the loadings of these different features towards principal components, how much variance do they explain. These are the metadata, x variable and y variable, these are stored, and how many components we have, right. So, here if we remember, we have 45 PCA components, and the variance that we have seen is this, right. So, PC1 is 22.9%, PC2 is 10.6, and so on. Okay. So, this is something here is kind of we can explore a bit more formally. So, for example, we have names P, right. This is again we explored with P dollar sign, right, and we can have something like names P dollar loadings, right. So, this is the principal components that we have, and what we can do is we look into the row names of P dollar loading. So, P dollar loadings, the column names are given as PC1, PC2, etc. Row names are given as these features, right. These are list of features, we have 45 features in the data. So, we can now look into this P dollar loading 1, right, and these are the values of these features towards that are loading the principal component 1. Okay. So, we can now more formally identify which are the features that kind of gives that has the highest loading for principal component 1, right. Why are you focused on principal component 1? Because this is the principal component that explains the highest percentage of variance, the most variance in the data. Okay. So, what we are doing is we are storing this as a matrix in this PC1 loadings, right, PC1 LDS as matrix. This list that we are getting, we are storing this as a matrix, right. And what we will do is we are going to change these column names as value. Okay. So, let us look into the PC1 LDS. Okay. And what we have column name as values, and these are the values of each of the 45 features, right. So, these are the loadings of each of the 45 features towards principal component 1. Okay. And what we are going to do is we simply take the row names of P loadings, right. What are the row names of this? These are the feature names, right, and see we will set those row names for as the row names of this PC1 LDS matrix. Okay. Okay. So, this is what this command means, right. Row names of PC1 LDS, this set from the row names of P dollar loadings, right. So, row names of P dollar loadings were the names of the features. Okay. So, we can again look into PC1 LDS, right, and as you see now these are the values we have. So, the column name is value and these are the features. So, this feature DMYPD, its contribution towards the PC1 is 0.012. Okay. So, these are the loadings. Now, again what you can do is we can sort, right, and identify which one of these features is the most important one for the PC1. Okay. So, here as you can see this parameter named TAIF50 that has the highest contribution towards the first principal component. Okay. So, that is what this means. Then you have TIF40, TIF75 and so on, and this is how the whole thing is organized. You might ask, like, can we look into the PC loadings of the second one? Of course, we can, right. So, for that we have to do a similar thing, right, as we did is we can create this PC2 loadings, right, and it will come from P loadings 2, right. So, what we can do, let us look at this. This is the head, right. So, the column 2 will give us the PC2 loadings. Okay. So, PC2 loadings we have loaded, right. So, here, we store that in PC2 loads. Okay, and then we can again okay. So, we are changing the column name, and the row name is again the same way for the PC2 LDS. P dot loadings. Okay, so all right, and then now we can sort this. Because this is a matrix, so we have to use this comma 1 and then decreasing class to true. Okay, and similarly we now know this feature, now this parameter num ss5 utr, this has the highest contribution to this feature. So, highest positive contribution of course, but you can also look into the negative contribution of these features, right, towards certain to this principal component 2. So, the best, if you want this, you can also look into something called the sort absolute value, right. This is the absolute contribution, and in that case you will see this minimum en ss that has the highest contribution in terms of absolute value. Of course, the direction will change, right, positive, negative, but this has the highest contribution. Okay, so this is fine. So, as you see, right, in PC we can now identify these features that has the most important role in the loading these principal components. So, the next step now is to actually visualize this, right, these results. Okay. So, the first thing we are going to do is look into the script plot, okay, and this script plot is using this function, right. Again, you can look into this script plot functions in the package manual, right. So, we have script plot P, that is the principal component analysis that we have done, axis lab size, title lab size, right. So, label sizes are given now, and here is the script plot. Okay. So, what you get is the principal components here, okay, and you have 45 principal components, and here is the explained variation in the data. As you can see, PC1 explains about 22%, 23% that we have seen. PC2 is about, I think, around 10-12%, right, 10% maybe, and this, as you can see, and this gradually decreases, right. So, this is expected. The red line gives us the cumulative variation in the data, right. So, again, this is something that is useful. We might want, right, how many PCs actually can explain up to, let us say, 80% of the variation or 90% of the variation in the data, and then maybe we want to look into these genes that actually load these PCs, right, and maybe those will be useful for prediction of different things here. Okay. So, we have now looked into the script plot. We can save this, right. So, we are going to use these commands. This is something we have already introduced and we are familiar with, and we are going to maintain this quality. Okay. The next thing we are going to do is the biplot, right, and here we have again the P, that is where the PCA results are stored. Show loadings, yes, we want to see the loadings, right, sorry, and here is the point size, side loading names, right. This is something is just kind of for the most of this is for aesthetic purpose, right. What we have is P show loadings true, point size. So, point size means point size of this each data points, right, that we have. Okay. So, remember these are samples. So, these data points are genes, right. For each gene we have data. Side loading names, right, the loading names, what should be the size of this text that we have. Color by group, right, this group has been already defined in the metadata, right. This is something I was referring to earlier, that we can use this information and use and color the samples, so that we can kind of see whether the principal components can separate out these samples well or not. legend position, right. So, this here is we have the legend, right. This is coming from the metadata. Again, genes are classified into different groups. This information is here, and n top loadings 15, right. So, 15 top loadings are shown here, and what you see here, right. So, let us now look into this plot carefully, and what we see, right, that is something that is important for our understanding. Okay. So, PC1 is fine, 22.8% variation, PC2 10.6% variation, etc. So, there are two things we can see here. One is about the clustering of the data points, right, and the other thing is about the variables, right, the features that you see, their directions. Okay. This is something that is probably again we can infer from here. Okay. So, let us say, so let us first look into the clustering of these groups. Okay. So, as you can see, like if you just very kind of carefully look into the, okay. So, what you can do maybe to what would be better is like if we switch off the loadings, right. Okay. And this is something that is helpful for looking at the clusters, right, and then we will look into the loadings a bit later and the variables sensor. So, what you see now is these purple points, right, they are kind of separated out from blue points, right. So, purple points, most of them are towards the left, and then you have blue points mostly towards the right. Okay. So, purple points are group F, right, GRF, as you can see, and these blue points are mostly group EM, and then lighter shade of blue is group EL, right. So, this is something that is interesting, right. So, what it means is that PC1 is kind of distinguishing between mostly group F and group EM points. Okay. And this is something that is what is clear here. For the rest of this cluster, it is not so clear, right, because this kind of they fall all over the place. Okay. These clusters are not separated very well by PC1 and PC2. For PC2, what you see is that it is kind of identifying a different cluster of blue points that are lying above these clusters here, right. So, it is kind of identifying a second cluster of blue points, okay, which is separated well by PC2. But PC1 is mostly differentiating between group F and group EM, right, that is purple and blue. Rest of the groups are not well separated, they are kind of all over the place, they are like overlapping with each other and along with the purple and blue. So, this is where we can do a second analysis, we can kind of explore other PCs, right, and see whether there are other PCs that will separate out these different groups, maybe group A, group B, or group A, group C, those kind of separations. Okay. So, which we will do a bit later. We can now add these loadings on top here, okay, and as you can see, right, these loadings, these variable names are here. As before, we have some of these variables that are kind of parallel to PC1. So, that means they have very high contribution towards PC1, and then you have some variables that are kind of perpendicular to PC1. So, they do not have much contribution, right. So, if you remember, it was around KI 40 or 50 that had the highest contribution for towards PC1, right. So, these are actually here around these arrows that you see. Now, that is something we have already discussed, right, in biplot when we discussed for other tools. What is unique here and interesting is because we are working with a variable like DMYPD, and this is something that we can consider, right, as a response variable or a dependent variable on other features, we can now start looking at the relationship between this feature and the other ones. Okay. So, for example, what you see that DMYPD is the direction of DMYPD is actually exactly opposite to the number of basic stretches, okay, here this one, or it is more or less in some similar direction as median promoter shape score. Okay. So, what this means, if you can see this, is that these features are likely to predict DMYPD in a predictive modeling approach. Okay. So, if you take this data, if you want to build a predictive model with linear regression or some other model like machine learning, DMYPD is likely to be predicted by these features, right. These are the features that might explain DMYPD much more. So, the features that are in same direction or similar direction or in opposite direction, exactly opposite direction, they are likely to be able to predict this DMYPD value. Okay. They are likely to be good predictors for this target variable or response variable. The features that are mostly perpendicular in direction to this feature they are likely to have zero contribution or almost no contribution towards prediction of this value. Okay, so this is something that this kind of information could be very useful if we are to build a predictive model in the next step. Okay, so this is something we can gather from this plot. One is of course the coloring part, the clustering of the points, that is something we can infer. Okay, this PC1, PC2 is able to separate out mostly purple and blue points, blue clusters, but not the other clusters. And the second is we can now understand the relationship between different variables, and in case you have a like target variable that you want to predict, right, we can look into the relationship of that target variable or response variable with other features or independent variables. Okay, so this is something that could be useful. All right, so let's move on now, and we'll use these plot loadings. Okay, and again all these functions are explained pretty well in the package. So you can see this, what is happening and what we are going to do. Okay, so I think very briefly I mentioned, right, we have plot loadings where we are now visually looking at the loadings of these principal components by different features. Okay, and here are the values, this color coding is here. So we are looking at the loading for first 5 principal components. So this is what is mentioned in this, right. So plot loadings, P range within this, label size, title, right. So this is the title of this plot. Then we have the subtitle, right. We have PC1, PC2, PC3, PC4, PC5, top 1% variables, right. We are taking only top 1% variables here. The color scale, right, that is the range that we are using, right. Those are blue, gray, orange, right. And the draw connectors is true. Okay, so draw connectors within that. Okay, so here are the labels, as you can see, for each of these features and the loadings. Okay, so if we kind of zoom into it, for 5 principal components, PC1, PC2, PC3 and up to PC5, top 1% variables, right, only top 1% are shown, not all of them. And what we see is like which features these features actually contribute the most or load the principal component 1 the most, right. And for PC2 we have seen some feature that has very high contribution, positive contribution, and then some features that have very high negative contribution, right. So similarly for 5 features, for 5 principal components we can see some of these features. And again, as I said, this is something that is very useful if you want to build a predictive model later on after this principal component analysis, right. When you started talking about principal component analysis, as I said, right, this is something that helps us explore the data and understand the relationship between different features that we have. This cannot help us predict directly, right. It does not build a predictive model here where we can predict something based on the features that we kind of identify here. This is something that we have to do separately, but this is a very useful exploratory data analysis, and some of these results can guide predictive modeling in the next step. The other important plot that we are going to do is something called pairs plot, right. As I said, right, and we have seen PC1, PC2, they are able to separate out only purple and blue clusters, right. Can we explore other principal components and see whether any of the other pairs of principal components can explain or separate out these other red, orange clusters, etc., right, or red, green clusters. Okay, so for doing that, we'll use these pairs plots, right, and this pairs plot command is like we are using this P, this is where the PCA results are stored, components get components up to, so 1 to 5, like PC1 to PC5, we'll plot first, and then here's something like just the kind of the aesthetics that is given, and color by group. Okay, so this color by group is very important because we want to see whether our analysis is able to, or some of the principal component pairs are able to separate out different groups, okay, that are actually shown by different colors. Okay, so let's look into the pairs plot here. Yeah, so it will take a bit of time, as you can see. All right, so here you see this. Okay, here is the pairs plot for first 5 principal components. Okay, PC1, PC2 we have already seen, right. This is something that we plot. Then we have PC1, PC3, PC1, PC4 and so on. Okay, so this is something what is showing us, right, these pairs of PCs, right, that are kind of the distribution of points if we look into the data from different combinations of principal components. Okay, so we have PC1, PC2, PC1, PC3, and so on. I think what you probably notice is like most of these is, of course, PC1 is separating out these purple and blue clusters, whereas PC2 is kind of restricted to separating out only these ones, right, these blue ones here, but they are not able to separate out any other clusters, right, maybe except this pair perhaps little bit. If you look carefully, maybe PC4, PC5, there is some sort of green ones are towards the right. Okay, so maybe we can explore the next 5, right, next 5 clusters here and see what happens. Again with a pairs plot. So if we want to say, let's look into 5 between 5 and 10, PC5 and PC10. So we'll use this pairs plot function again, right. Okay, so this is now creating this pairs plot function. Okay, so here now we have these pairs, right. Here it's PC5, PC6, you have PC5, PC7, right, and here you have PC6, PC7, and so on, right. So you can see these pairs. What is interesting, as you can probably notice here now, is, for example, PC5, PC10, right. It's kind of separating out these green and orange things, right. So you can see these green clusters above here and orange clusters are here, right, below here. So this is interesting because then if we want to separate out this cluster, it means PC5, PC10 might be the most informative principal components. Okay, so this will happen in many cases in your data, right. You will see some of these clusters can be separated out by other principal components, not by just PC1 and PC2. Okay, so let's choose this PC5, PC10, and maybe we can do a biplot and see, right, the PC5, PC10 analysis, right. And we can generate this biplot with this principal component P. X would be PC5, Y is PC10, color by group, right. We are keeping this color group. H line, V line equals to 0, right. This horizontal line and vertical line. And legend position, where the legend position should be, show loadings true, and these are kind of the end of loadings 20, Y limit, etc. All these values are given. So maybe we can simply write this for now. We can write we can run the whole thing, and then we can see, right, which whether we can make it a bit cleaner, right. So let's okay. So for now we can make the loadings false, right, and you can simply look at the colors. Okay, and this is something interesting, right. If you can see this color, and of course you can do this hands on yourself and you will see this. What we now notice is that these orange and green clusters, they are kind of separated out by these PC5, PC10, right. So the x axis is PC5 and y axis is PC10, and these green and orange clusters, they are separated out very nicely by this PC5 and PC10, right. So PC5 is probably the one that is actually separating out these two clusters, right. So maybe this is something that's very also again informative, right. So what we saw that purple and blue, they were separated out by PC1, right. So what we want to do is look at the loadings of PC1, what are the features that are loading the PC1, right, the top 1% or 2%, and these are the features that will be able to predict the kind of explain the difference between our purple and blue cluster. Similarly, if we look into the PC5 loadings now, we will be able to differentiate between the green and the orange clusters, right. So this is something that we can explore and see, right, different principal components, and that means different sets of genes can explain or explain the variation in the data and kind of reveal the structure in the data, and they might be useful for predictive modeling later on. Okay, so with that we have completed the hands-on. So we will go back to the presentation here, back to the, so we will summarize. So we have now done PC analysis using PCAtools as well, right. We started with PRComp tool, then we use VectorMind R, and then finally we used PCA tools, and in all of this we have seen the biplot loadings in some way, right, and we could sort these loading values and we could identify the most important features, whether genes or some of these features that we talked about in this data set. In PCA tools we have talked about something unique where we can look into the pairwise plot of PCs, right, and this might help us identify the best set of PCs for separation of these different clusters, right. Not all clusters of data or not all sample types can be separated out by PC1 and PC2. So, there would be other PCs sometimes that will be more informative for separation of these data points or data clusters. So, this concludes our discussion on principal component analysis, and then next in class we will be talking about UMAP analysis, and we will be also doing a hands on that. Thank you.